Today I'm going to be modeling mitosis, and I know mitosis is something that you've covered at some point in the past, and it feels really familiar, but we need to pay attention to the details. And one of the things that's really important is that we know the definition of the vocabulary, so please go back and review the lecture slides. Make sure you know those terms. A lot of our terms sound very similar but have different definitions, and it's crucial that we understand what those terms mean and what they are referring to. So we're going to follow one cell as it goes through mitosis cell division and makes two genetically identical cells. What I've modeled here with this cell is the cell membrane in orange, the nuclear membrane surrounding the nucleus with DNA inside in green, and these blue chunks over here are our centrioles. Now remember, most of our cells are going to spend most of their time in interphase, reading and expressing their genes. So things like stomach cells will spend about 2 days in interphase, reading and expressing genes before they get that signal to divide. And cells like neurons will spend potentially their whole lifespan, or our whole lifespan as a human, in interphase reading and expressing genes. They would not have received the signal to divide since we were about 2 or 3 years old. So let's say our cell here has already received the signal to divide, and it's entered into those later stages of interphase. It's entered into G1. Now during the G1 phase, this cell is going to start to expand. It's going to get bigger in size as it makes more cytoplasm, as it makes more rough and smooth endoplasmic reticulums, more lysosomes, and it starts to double all of those organelles. Now unfortunately with chalk and cement, I can't accurately show that, but in your mind, just imagine that the cell is getting bigger in size. Inside of our cell, we have 3 pieces of DNA. Our DNA is in the form of chromatin. It is loose. The genes are being expressed. Once our cell has been expanding in size, there is a checkpoint. And essentially what's going to happen with that checkpoint is the cell is going to ask itself, do I have enough resources to divide? This is especially important when we think about organisms that may be near starvation. If a cell doesn't have enough resources to go through this really energy-intense process, then it shouldn't. It should stop right here. Don't even try to invest that energy. But let's assume our cell has enough resources to divide. It's ready to enter into the S phase. During the S phase, our cell is going to go through replication. Now we covered replication. Replication in chapter 11 talks about with one piece of DNA, helicase first unwinds the DNA, DNA polymerase will build the new strand, and DNA ligase will fuse together the sugars and phosphates of that new strand, and then telomerase will add the telomeres at the end. The process of DNA replication, making an exact copy of each piece of DNA, can take upwards of 11 to 12 hours in a eukaryotic cell. This takes a lot of time. Once our cell is done copying all of the DNA during the S phase of interphase, it will enter into the G2 phase. During the G2 phase, our DNA is going to get condensed into chromosomes, and our cell is going to make the sister chromatid structure by holding the copied DNA together with a rubber band of proteins. And remember, that rubber band of proteins is called the centromere. So what I am modeling with my sock pieces of DNA is I am modeling that formation of the sister chromatid structure. Now the reason why this structure is so important and why we use this terminology is because the rest of cell division are mitotic M phases, prophase, metaphase, anaphase, telophase. This is all about moving the DNA. If we screw it up, if the new cell gets an extra piece of DNA or is missing a piece of DNA, that cell will be non-functional. We're going to have to kill it. We're going to have to send it through apoptosis, cell death. It will not be able to perform the function. So it's absolutely crucial that we move the DNA, we don't leave any pieces behind, and that both of our cells at the end are genetically identical to our original cell. During the G2 phase, we have another checkpoint. Our cell is essentially going to be asking itself, did I copy all of the pieces of DNA? Let's assume that the answer to that question is yes. We're going to enter into our mitotic M phases. The first mitotic M phase is prophase. During prophase, our cell is going to get rid of the nucleus. The whole point of getting rid of the nucleus is to make sure that we can move the DNA, so we have to get that structure out of the way, which is why the green is now broken into little chunks. Microtubules are going to attach onto our centrioles and reach out to catch our sister chromatids. During metaphase, our microtubules are going to line our sister chromatids up in the center of the cell. Metaphase is very distinctive to see underneath the microscope. We can clearly see all of the DNA lined up in the center of the cell. During anaphase, our microtubules are going to tear our sister chromatids apart, and they're going to move the individual chromosomes to opposite sides of the cell. Now, when I do this in the classroom with the dry erase board, it's really easy to erase those microtubules. Unfortunately, with chalk, it doesn't work as easily. So we tear apart the sister chromatids, and the microtubules move those individual chromosomes to opposite sides of the cell. During telophase, a new nucleus will form around our DNA. Our DNA will start to unwind, these individual chromosomes will start to unwind into chromatin. So here we have in green that new nucleus. Our DNA is unwound into chromatin. Cytokinesis will happen to separate the cytoplasm and all the organelles to the two separate cells. At the end of cytokinesis, we will have our two separate but genetically identical cells. As always, if you have any questions, please email me